computational biologist. All right, thank you very much. Yes. All right, thank you very much. It's a pleasure to be here. My lab is interested in the question of how sequence and structure impact function in proteins. And we'd like to translate this understanding into effective tools for protein design. Of course, I think that protein design is the most complex, or one of the most complex at least, and multi-dimensional problems in molecular biology. And because it's such a complex and multi-dimensional problem, the theme of my talk will be that an effective strategy to deal with this complexity is to combine inferences from multiple disciplines, including bioinformatics, atomistic design calculations, and machine learning and AI. So whereas each of these disciplines on its own is not really able to deal with this complexity, the combination is very effective. So to provide some sort of a practical motivation for computational protein design, of course, proteins are incredibly useful both in research and in applications. But as a very general rule, almost no natural protein is ever good enough for applications. For instance, their stability may be too low, even for lab use. The activity levels may be low, and even expressing proteins in common lab hosts, such as bacteria or yeast, may be incredibly challenging. And the underlying reason for all these problems in protein engineering and design is that proteins are literally the most complex molecules we know in nature, and that the majority of mutations, the vast majority of mutations to natural proteins actually lead to dysfunction rather than to gain of function. And because of that, essentially the paradigm that's being used in optimizing natural proteins relies on the winning evolution or trial and error. You start with some low level of activity, shown here as a sort of fitness landscape, let's say activity or stability of your protein. You introduce diversity, let's say by rational or semi-rational or irrational mutagenesis, and then we select from these variants the one that increases the desired property the most. But because one mutation never really carries the day, you have to repeat this process multiple times, and this leads to these samsaric cycles of mutation and selection. It can take years even, in some cases are completely frustrating and nowhere. But even if this process is successful, it often leads to these suboptimal peaks where one property, let's say the activity of the protein has risen, but another property, such as its stability, has deteriorated. And these are known as trade-offs, and they're incredibly frustrating. Again, sometimes they're completely unacceptable and can lead to stopping such a project. Our goal in protein design, okay, so let me just back up. These are practical concerns, of course. And in addition to these, of course, the fact that we still rely, 2023, AI, right, all these revolutionary technologies, the fact that we still rely on these iterative, irrational processes in order to generate life-saving drugs and enzymes for industry means that there are really serious gaps in our understanding of how sequence and structure encode for function in proteins. And our goal in protein design is to minimize these gaps and, if you will, enable something like this, a one-shot design where we encode all the mutations that we need in order to reach our goal, no trade-offs, no samsaric cycles of mutation and screening. So can we do that? Perhaps the most general problem in protein engineering design is the fact that most natural proteins are only marginally stable, and that means that often it's even very difficult to express them heterologously. So already a number of years ago, we developed this method called PROS, for protein repair one-stop shop, that enables stabilizing biologics essentially in one shot. Here, in this first example where we tried PROS, we targeted a human enzyme called acetylcholinesterase. It's a very important enzyme in neuromuscular junctions, never mind the details, but it's also a very, very challenging enzyme, 550 amino acids in length, multiple disulfide bonds, glycosylation sites, and even though this enzyme was the target of protein engineering for 2 decades before our work, there was really no variant of this enzyme that was shown to be more stable and more producible, more expressible in microbial hosts. So here we show that we designed essentially just 5 variants using a combination of evolutionary constraints and atomistic design calculations, as I'm showing on the top. We designed 5 variants. Each of them encode dozens of mutations from the wild type protein, which we introduced simultaneously to the protein. So in total, we tested just 5 variants. All of them exhibited a marked increase in thermal stability, 20 degrees, relative to the human variant. But the most dramatic effect came when we tested, very dramatic. What did they do? Oh, nice. Thank you. Maybe you should say this. The really dramatic effect came when we tested the bacterial expression levels of these designed enzymes. You can see that all the 5 designs express more than 100-fold, and up to 2,000-fold higher levels than the human enzyme shown on the left. The human enzyme barely expresses in E. coli. So this was very dramatic. Then many people were interested in this, so we developed this into a web server, and this has been used literally thousands of times since. There are more than 100 peer-reviewed publications and patent filings that show successful application of this method to various problems. Of course, all the methods that I'll show you depend on having a reliable structure. And this came up multiple times here. Is AlphaFold really reliable for that? My answer is almost dramatically yes. Here we showed the case where we took an enzyme family for which there are almost no published structures because these enzymes are so difficult to produce in lab hosts. These are enzymes coming from fungal hosts called unspecific peroxygenases. Very important for biotechnology, but very difficult to work with in the lab. We took 10 of these enzymes for which there are no structures because they're inexpressible. And for 9 of these, we designed variants that were highly expressed, stable, and highly active in common lab hosts, essentially Saccharomyces cerevisiae or Pichia pastoris. So this shows an approach for enzyme discovery, accelerated enzyme discovery. I think it's very exciting. We've also applied such approaches to antibody design. Often in antibody discovery campaigns, you have the sequences of the antibodies, multiple antibodies, but you have no crystal structures for them. Current AI-based structure models for antibodies are quite accurate in most of the antibody, not in H3, of course, and that enables also reliable computational design. We've also recently extended PROS and other methods from the lab to member proteins, and here I'm showing their application to voltage-gated potassium channels on the top, and to a membrane, human membrane enzyme. Both of them are very difficult for heterologous expressions, and they rose by 10-fold approximately, using, again, one-shot computational design. The second one, this ceramide synthase enzyme, again, no crystal structure. We relied on an AlphaFold model for it. So that's great. The reason why this is, I think, very exciting is that stability, the marginal stability of proteins has been considered to be really a very long-standing problem in protein engineering and design, almost 30, 40 years. And this shows that the fact that the same strategy can address multiple problems in protein stability design shows that the stability design principles that we encode in PROS and other methods from the lab may be quite general and may extend to all proteins. So of course, the complementary question to stability is whether we can also improve activity. This is a method, again, from a few years ago called FuncLib for improving the catalytic rate or specificity of enzymes. Here, again, the first target we looked at was an enzyme called phosphotriesterase. This is a natural enzyme, bacterial enzyme, that breaks down toxic nerve agents. These are some of the, actually, the most toxic to human compounds we know, synthetic compounds. And in this case, we showed that other than the natural enzyme, it exhibits some activity against these toxic nerve agents, such as VX, Russian VX, etc. Its activity levels are far too low for therapeutic use, whereas 4 of the designs that we generated here, essentially within a few months of starting the project, were between 100-fold and up to 4,000-fold greater activity than the wild type, essentially reaching the therapeutic needs through one-shot design and experimental characterization. This is also available as a web server. And these web servers are being used quite extensively, and this has allowed us it has demonstrated that the methods are applicable to of very wide range of enzyme classes, therapeutic proteins, including vaccines, and other types of antibodies, etc. So that's very exciting. When we saw that these evolution-guided atomistic design, this paradigm that we're developing, this combination between bioinformatics and atomistic design calculations, is so effective and is largely automated, we asked whether we can push the limits and explore designing very large repertoires of functional proteins, including antibodies, enzymes, and other types of proteins. And this combines machine learning and AI. So here in this project, we asked whether we can design literally millions of different potential enzymes by designing small fragments of enzyme structures such that they would modularly combine with one another into millions of different designed enzymes. Of course, this was unprecedented at the time, but we were inspired and sort of encouraged by the fact that modular assembly is actually used in nature in order to generate highly versatile proteins. In this case, as you can see here, this is essentially the principle by which antibodies are generated in our bodies. Of course, our bodies are able to generate billions and billions of different antibodies, but there's no space for encoding so many antibodies in our genome. The way this is done in nature is by recombining these genomic cassettes, what's known as VTJ recombination. So there are a few dozen of these cassettes in the genome. They're randomly combined to generate antibodies, and those are selected. It's an amazing property that by random recombination of these genomic cassettes, you get often, of course often not, a stable, foldable, and functional antibody. But this versatility, we don't know of another parallel system in nature that generates versatile protein functions in this way. We absolutely don't know of any enzyme system where versatility or diversity is generated this way. So here in this Cadence project, we asked whether we can apply the same principles, go into an enzyme family, break it down into pieces, design the pieces for modularity such that they would recombine with one another to generate a mostly folded, stable, and potentially active protein, as shown here, and then screen this experimentally. So very briefly go through the motions here. It's a quite complex approach, but we started here with an enzyme family called glycoside hydrolase 10, xylanase family. There are about 80 structures in the protein data bank for this enzyme family. They exhibit diversity in the structures, as you see here. We break them down into 4 pieces, use PROS in order to design more stable fragments, and FuncLib in order to diversify the active site pocket. And then we applied the new machine learning method that we developed in order to select those fragments that are predicted according to atomistic design calculations to combine with one another and to form low energy structures according to atomistic design calculations. Then we simply go to the lab and synthesize these fragments and recombine them, fuse them using Golden Gate assembly. Never mind the details for this. Essentially, we're generating fusions between all these fragments, generating millions of different enzymes in the process. We then use an activity-based probe, shown here, and provided to us by Herman Overklett from Leiden University. This activity-based probe mimics xylan. The chemical moiety shown here is xylan, but instead of being hydrolyzed by a functional enzyme, the enzyme covalently attaches to this, and you can see that it's also fused to a fluorescent probe, and that means that we can then use yeast display and FACS sorting in order to select the functional population from the design population. Then we use deep sequencing in order to detect what are the functional enzymes, and of course our interest is not just to produce many different xylanases. Our main interest is to learn new enzyme design principles. So we've trained a machine learning method to discriminate between the functional and non-functional enzymes in this set. And then, if this machine learning predictor is successful, then we should be able to return and generate a second generation repertoire that will exhibit higher rate of functional enzymes. And this was indeed successful. In the second generation, we produced 10-fold more functional enzymes than in the first one. In total, in this procedure, we generated, in 2 such selection rounds, we generated more than 15,000 active and diverse enzymes. How diverse are they? They exhibit between 80 and 160 mutations from any natural enzyme in NR, right? So these are very, very different from natural GH10 family enzymes. And I'd like to really underline here that this diversity is not limited to just sequence changes on the surface that don't impact function. They really change the shape and electrostatics of the active site pocket. And that meant to us that potentially we will see functional differences among these designs. We asked Herman Oberkleff for another activity-based probe. So on top we see the xylobio's xylem mimic. On the bottom we see a cellobio's. This is a cellulose mimic. You see it's a bit different in chemical terms. And in the second generation repertoire, we selected both xylobio's, xylamases and cellulases. And you can see that we're getting cellulases. Out of this, we're getting more than 9,000 xylamases and more than 3,000 cellulases, and there's only a small overlap between them. So we are actually seeing functional differences or orthogonality, some sort of orthogonality in this library. And this is encouraging that this sort of strategy may be able to discover new activities that don't exist in nature, of course, with other probes, for discovering new enzymes with desired properties. So when we saw that this sort of repertoire design strategy successfully generates highly functional repertoires, we wondered whether this could also be applied to more biomedically relevant problems. And here we developed a method which we call LaFAD for generating affinity-improved antibodies, starting from an antibody-bound structure. And here the question is, so similar to the in vitro evolution studies that I mentioned in the start of my talk, also when you look at affinity maturation, either in nature, in our bodies, let's say, or in the lab, these maturation strategies are incremental, right? We add one mutation at a time, again, they're very tedious, but they also lead to these trade-offs between stability and binding affinity. And often when affinity is pushed to the limit, let's say an antibody that increases affinity by orders of magnitude, you often observe a decrease in stability or expressibility in conventional affinity maturation techniques. Here the idea was not to generate yet another maturation technique, but to generate a library, one-shot library, from which we will find antibodies that will increase the affinity while hopefully not exhibit trade-offs with stability. So to do this fairly quickly, we take an antibody-bound complex. In this case, my favorite antibody targets lysosome. Nobody else cares about this antibody, but it's very easy for us to work with in the lab. We targeted 9 positions on the CDRs. This is really the interaction surface complementary to determined regions. The surfaces that interact with the antigen. And we designed a combinatorial library using the strategy I showed earlier, cadence, and another strategy I didn't mention before. In this library, none of the mutations is chosen randomly. All of them are chosen according to our optimistic design calculations to improve the affinity, predicted to improve the affinity for the antigen. But crucially, they're not just chosen simply, they're chosen to be compatible with one another, such that they wouldn't clash, they wouldn't form negative epistatic interactions between them. So then we generated a phage display library. This is a small library, but we still went for phage display. And we selected more functional, higher affinity variants from this library. We were encouraged to see that after selection, the library seemed to completely wipe out the wild-type antibodies starting point, potentially meaning that that the entire library was very enriched for higher affinity variants. Not only that, you can see that the variants encode between 4 and 8 mutations from the wild-type antibodies, something that you don't normally see from one round of affinity maturation in antibodies. Then we took 4, selected 4. Out of these, there were thousands of antibodies that were selected according to deep sequencing. We chose just 4 of them, expressed them, and tested them using SPR. And what you can see here is that the starting antibody exhibits about 3 nanomolar affinity for the antigen, for lysozyme, and we improved it by 1,000-fold using, again, one-shot library design. So next, of course, we asked whether these antibodies exhibit trade-offs with expressibility and stability. The answer is no. They express better than all the 4 designs we tested express better than the wild type, and they're also a little bit more thermally resistant and really coming pretty close. Of course, we'd like to learn rules for effective design also in small scale. Yeah, no trade-offs. Okay. We'd like to learn rules for effective design also in small scale. Here we collaborated with Mark Craig's lab in Southampton. In this case, library screening was not an option. He told us they can only test 10 designs. We designed 10 antibody variants, starting from a completely different antibody targeting a cell surface receptor. And you can see that we improved affinity to below the nanomolar range, which was critical for their experiments. This is my current obsession, recently approved for funding by the ERC, where we asked the question, can we, instead of fixing defective antibodies as we've done so far, can we go to the source of the problem and design an excellent repertoire of human antibodies, including billions of different human antibodies that are stable by design. Using our design strategies, we'll generate phage display libraries using this, and then provide them to anybody who wants to screen for new antibody binders. But crucially, this will also allow us to learn antibody design principles and hopefully accelerate drug discovery. With that, I'll close. I'll mention, again, circling back to the point I started with, many people, or many companies even, are now approaching the problem of protein design using sort of a monolithic AI-based strategy. But these strategies are not reliable enough, as I think we see in papers. What I've shown you here is that combinations of inferences allow for very accurate control over protein stability and activity. We typically don't see trade-offs between activity and stability. And what I'm most excited about are these more high throughput or large scale repertoire design strategies. I think that this will enable not just optimization, but also discovery of new functions. So thank you very much.
Thank you. I didn't understand why you call all natural proteins suboptimal. I mean, when you consider all their various functions and environmental changes in which they have to work, I think they are pretty optimal. And when you take, for example, the category of stability, some proteins are designed not to be stable, like transcription factors, and they have especially the IDR regions in them not to be stable. So why do you call them suboptimal?
No, I completely agree. Natural proteins are optimized to be good enough for their natural host, and they've undergone millions of years of optimization to that end. The problem is that human use is quite removed from our needs are different from what we observe in nature. So for instance, if you'd like to use an antibody, let's say an antibody that's very potent against some new virus in our bodies, it can neutralize many different strains of this virus. If you want to develop this into a drug, this antibody needs to be stable for 6 months in a test tube. That's something that our B cells don't care about, so they don't optimize for these properties. Similarly for enzymes, vaccine immunogens, etc.
So, Sarel, how do you pick 4 antibodies out of 1,000?
Yeah, it was largely. I didn't think this would work. We were not expecting large improvements here. So we basically chose ones that seemed to be different in sequence enough to be interesting for us. It was out of the deep sequencing. We knew that these were enriched, and we just chose ones that were different from one another. But there were thousands of antibodies that were enriched in this deep sequencing experiment. It was really a shocking result. I think there may be others that are even better in this sector.
Last question. So, what do you think would happen if you take one of your super enzymes and stick it back into the organism that actually expresses this?
Right. I think this goes back to the previous questions. What is optimality? Right. Well, what sort of optimality do we mean? We typically talk about optimality in biotechnological or lab use case, not in the organismal sense. I can say that this is not being done very much, but there's one paper that came out. We had a collaboration with Tony Fleishman in our department who works on neuronal Gaucher. It's a very severe disease. I don't know anything about it. But the idea was he tried to use one of our designed I think it's called beta-glucose ribosylase. This is an enzyme that's mutated in the normal Gaucher as a model mouse for this. You made an AAV and in same words here that you would understand. And it showed that the design variant, which is more stable, really reverses the effects of the disease, whereas the Sarel, which is currently used as a therapeutic against this disease, has very little effect. So the mice are happy after 200 days. But that's it. Again, it's more. I think this still is more in the biomedical.
Yeah, it's not really a fitness answer.
I agree. Yeah, it shows that even in the body these are tolerated. But I agree. Whether these things, design proteins, can be implemented in an organism, and do they actually provide some benefit? We don't know. This has not been done.
You?